Hello everyone, today we are going to discuss biotechnology applications chapter based on CBSE term 2, and we will cover complete theory of covering NCERT and case based questions as well as subjective short long questions, and we'll try to complete this chapter in 20 minutes or less time. So let's start. In this chapter, we are going to study by applications in agriculture, medicine, transgenic animals, and ethical issues. So, what is biotechnology? How it is useful? It is useful to human beings to produce micro, plant, animals, and metabolic machinery. And this is possible because of recombinant DNA technology only, which is valuable and useful manipulation of the genes on the large scale and up to obtain the desirable products. Okay, then we come about GM, genetically modified crops, and therapeutics, diagnostics, agriculture, processed food, bioremediation, etc. Now, the areas of biotechnology are to provide the best catalyst in the form of improved organism, means a pure enzyme, genetically modified crops, and these all are the various applications of biotechnology. And what are the basic areas on which critical research is going on is that getting a pure enzyme, creating the optimal conditions, and the most important is downstream processing. Biotechnology applications in agriculture. Now, to feed the large population, as the population is increasing day by day, we need to increase the food production. So this can be done only by using agrochemical based agriculture, organic agriculture, or genetically engineered crop based agriculture. Now, the point comes about green revolution. What is green revolution? It is the revolution which increases the yield of crops and by the use of agrochemicals and the better management practices, and the food production has increased to threefold. Okay, then most of the agrochemicals means agriculture based chemicals or chemicals derived from agriculture, so they have harmful effects and they are also expensive, so and it is difficult to further increase the yield with the existing varieties also. So the solution for these agrochemicals, which are costly, and these conventional breeding methods which are difficult to increase the yield, now we have developed a new method, that is genetically modified organism, or you can say GM crop. We have studied further in the introduction. So let's start. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are known as genetically modified organism. Okay, and the genes, now these organisms whose genes are altered are known as transgenic organisms, and since they have foreign gene, now they are known as transgenes. Now, how they are useful? Basically, crop become tolerant to the abiotic stresses like cold, drought. Dependence on chemicals has reduced. Post harvest losses are reduced. Efficiency of mineral usage has been increased, so prevent early exhaustion of fertility of soil, means efficiency has been increased for minerals. Nutritional value has been enhanced, means vitamin A, enriched rice come now, golden rice, okay, they have vitamin A nutrition. Biopesticides, application of biotechnology in agriculture is production of pest resistant plants. Biopesticides means using living organism as a pest resistant plants would decrease the amount of pesticides, and Bt toxin is produced by bacterium called Bacillus thuringiensis. Bt, Bacillus thuringiensis toxin. This gene has been cloned from bacteria and been expressed in plants to produce resistance to the insects without the need for insecticides, and so named as biopesticide. For example, Bt cotton, Bt rice, Bt corn, tomato, potato, soybean, etc. Now, what is Bt cotton? Very important. You can expect one question from genetically modified organisms and Bt cotton. Bt cotton means it is a strain of bacteria Bacillus thuringiensis. What does bacteria do? Bacteria produces a protein. That protein kills insects. For example, Lepidopterans. This insect is killed. Tobacco budworm, armyworm. These are known as Lepidopterans. Coleopterans are known as beetles, and flies and mosquito come in the category of Diopterans. This forms the crystal. This bacteria form a protein crystal during the growth phase, and that crystal contain a toxic insecticidal protein, but it doesn't kill the bacteria because Bt toxin protein exists as inactive protoxin. Basically, अगर आपके दिमाग में क्वेश्चन आएगा, you have question raised in mind that if this Bt toxin can kill the insects, that why do not this kill the bacteria itself? Because the Bt toxin protein inside the bacteria exists as inactive protoxin. But if the insect ingests it, if the insect intake it, then it converted into active form due to the alkaline pH. That is why the bacteria kills the insect, not itself. The activated toxin now, what it will do? It will create the pores on the midgut of the epithelial cells of the insect and will swell it. The cell of the epithelial cells will swell up and they will die because the pores are created due to death of an insect. So Bt tobacco was first cultured to kill the ornworm Manduca sexta. Its scientific name is Manduca sexta, and this toxin is encoded by the cry gene, which are insect group specific. For example, cry1ab kill corn borer, cry2ab kill cotton bollworm, cry3ab kill Colorado potato beetle, then cry3bb again corn rotworm, cry1ac kill cotton bollworm. Let's revise this. Corn 1AC and corn 2AB kills cotton bollworm. Corn 1AB, corn borer. Corn 3AB, Colorado beetle, potato beetle. Cry 3BB, corn rot worm. Very good. Now applications in medicine is in therapeutic drugs. In the production of therapeutic drugs, about 30 therapeutic drugs, recombinant, have been approved in the world and 12 are marketed in India. Now, genetically engineered insulin. In the management of adult diabetes, insulin needs to be sufficient, available. But insulin is used for diabetes, which was in early stage extracted from the pancreas of slaughtered cattle, dead cattle, and the pigs. But it causes allergy because it is a foreign protein. That is why, firstly, let's understand insulin. So insulin has two chains, A and B, which are linked with disulfide bridges. In mammals, human insulin is produced as prohormone, means until and unless the hormone is maturing or being functional, it is synthesized before. And it also contains an extra stretch, this called known as C peptide, C chain. This is not present in mature insulin. This is removed during the maturation of insulin. Main challenge is to assemble the insulin in mature form. In 1983, mark the year, Eli Lilly, the company, American company, have prepared two DNA sequences corresponding to A and B chain of human insulin and introduced them in the plasmid of E. coli to produce the insulin chains. And these chains A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin called as humulin. So who created humulin? Eli Lilly in 1983, this American company. Now what is recombinant vaccine? The vaccines which are produced through genetic engineered methods are known as recombinant or second generation vaccine. They have genes insert for surface protein of pathogen which enhance the immunity but do not result in infection. These plasmids are inserted in bacteria that express the viral protein which are injected as a human host as a vaccine. Therefore, they are recognized as foreign and immune response is elicited. So, recombinant hepatitis B vaccine and polio vaccine are the rDNA vaccines. Stem cell technology. Now, in this time when we are developing so fast, we should know that the how to treat the various malignant and non-malignant diseases by stem cells. Basically, stem cells are undifferentiated biological cells that can differentiate into specialized. For example, my brain has some problem, so I can make the stem cells to grow there and take the function of the brain and produce more stem cells. So stem cells has the full potential to generate the full organ or tissue. For example, it is used in bone marrow transplantation, brain disease treatments, blood disease treatments, cell deficiencies, cardiovascular disease treatments, and all. There are two types of stem cells. One is embryonic, which is isolated from inner cell mass of blastocyst. When the parturition delivery is going on, stem cells are just carried out. They are taken out and stored in the stem cell bank. And the adult stem cells, which exist throughout the body after embryonic development, and they will be found in the brain like tissues. And after, if we have some problem in brain, we can grow the stem cells and use that inside it. Gene therapy. Gene therapy is replacing the defective gene with the functional gene, and it works effectively against the single gene disorder, enzymatic disorder, and immunodeficiency disorders. First gene therapy was performed in a 4-year-old girl in 1990 who has ADA deficiency. ADA is adenosine deaminase, and this enzyme is very much crucial for immune system to function. And this disorder is caused due to the deletion of genes for adenosine deaminase. So, in some children, AD deficiency can be cured by bone marrow transplantation or enzyme replacement therapy. But the problem with this approach is they are not completely curative. That is why gene therapy was introduced. Gene therapy provides a foolproof treatment. The steps of enzyme replacement gene therapy is: firstly, lymphocytes from the blood of the patient are grown in the culture outside the body. Then functional ADA, retroviral vector, is introduced into these lymphocytes. Now this ADA gene is introduced in the lymphocytes and it is returned to patient. And these cells are not immortal. The patient requires the periodic infusion of this one. But if the gene isolated from marrow producing ADA is introduced into cell at the early embryonic stage, it would be permanent cure. Like hemophilia, cystic fibrosis, Parkinson diseases can be treated by it. Transgenic animals whose genes have been DNA or genes have been manipulated, whose have transgenes or foreign genes. Example: transgenic rat, rabbit, pig, sheep. 95% mice are transgenic animals. Now, what are the benefits and uses of transgenic animals? First, normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functioning. For example, the study of complex factors involved in growth such as insulin growth factor. मीन्स ट्रांसजेनिक एनिमल्स को हम यूज़ कर सकते हैं, उनको ऐसे बनाया गया है, दे आर मेड सच दैट कि हम उसमें देख सके कि जीन कैसे रेगुलेट हो रहा है, जीन में कैसे उसकी फंक्शनिंग है, उसकी डेवलपमेंट कैसी है, जैसे इंसुलिन के लिए किया गया था. स्टडी ऑफ डिसीजेस, हम डिसीजेस को स्टडी कर सकते हैं जैसे कैंसर, सिस्टिक फाइब्रोसिस, रिमेटॉइड आर्थराइटिस, अल्जाइमर डिसीज, ठीक है, ये मॉडल की तरह इनको ट्रीट किया जा सकता है। बायोलॉजिकल प्रोडक्ट्स मीन्स जैसे कि ये एक बायोलॉजिकल प्रोडक्ट्स बनाने में काम आती है, जैसे डीएनए, जैसे हमारा ह्यूमन प्रोटीन है अल्फा 1 एंट्रीट्रिप्सिन, ये यूज़ किया जाता है एम्फीसीमा डिसीज़ को ट्रीट करने के लिए। और भी होते हैं जो फिनाइल कीटोनुरिया या सिस्टिक फाइब्रोसिस के लिए यूज़ होते हैं, तो इनको बायोलॉजिकल प्रोडक्ट्स बोलते हैं। सेकेंड एग्जाम्पल आती है 1997 में, जो फर्स्ट ट्रांसजेनिक काउ थी हमारी रोजी, जो कि ह्यूमन प्रोटीन एनरिच मिल्क देती है 2.4 ग्राम पर लीटर, ठीक है। जो मिल्क है उसका, उसके अंदर होता है अल्फा लैक्टा एल्ब्यूमिन, जो कि न्यूट्रिशनली बहुत ज़्यादा बैलेंस्ड है और ह्यूमन बेबी के लिए बहुत ज़्यादा ज़रूरी है। नेचुरल काऊ का मिल्क भी इतना ज़रूरी, इतना मतलब वो नहीं होता बैलेंस्ड न्यूट्रिशनली, बट दिस इज़ द न्यूट्रिशनली वेरी बैलेंस्ड। वैक्सीन सेफ्टी, जो ट्रांसजेनिक माइस होते हैं, जो हम वैक्सीन्स बनाते हैं ह्यूमन में टेस्ट होने से पहले माइस में या किसी भी मॉन्की में यूज़ कर जाती हैं। जैसे पोलियो वैक्सीन माइस में देखी गई थी, और जैसे मॉन्की या माइस में अगर सक्सेसफुल है देन ह्यूमन में डाली जाती है। केमिकल सेफ्टी टेस्टिंग, इसको कहते हैं टॉक्सिसिटी, मीन्स ड्रग कितना टॉक्सिक है उसको चेक करना। क्या करते हैं हम, उसमें टॉक्सिक सब्सटेंस जब हम डालते हैं, जो एनिमल्स होते हैं ट्रांसजेनिक वो बहुत सेंसिटिव होते हैं, उनकी बॉडी में कोई ना कोई इफेक्ट आ जाएगा। अगर तो टॉक्सिक सब्सटेंस जाने पर उनकी बॉडी में अगर इफेक्ट आया, दैट मीन्स द केमिकली इट इज़ नॉट सेफ, ठीक है। देन वी हैव इज एथिकल इश्यूज। एथिकल इश्यूज में क्या है, मान लो मैंने किसी पर्सन के जीन्स के साथ छेड़छाड़ की, I manipulated the gene of any human, तो लोग तो लड़ने आ जाएंगे मेरे साथ। People will fight that how you have modified it, who give you right if it gives unpredictable result, ठीक है। Therefore this modification needs the patent granting. So firstly we have many things in this. Firstly is bioethics, means we need to have some standards, ethical standards to evaluate our actions. What we are doing, our activities need to be regulated in this biological world. Public, because public will be concerned basically if the patent is not concerned, patent is not for any product. Therefore, in order to control these all issues, what Indian government has done, it has set up an organization which is Genetic Engineered Approval Committee, GEAC, which will make all decision regarding validity of GM research and safety of GMO for public services. Now patent कर रहे हैं. What is patent? Patent is the right that ये चीज़ मेरी है. This property, this biological entity belongs to me. If I am talking about biological entity, it will be biopatent. It is a set of exclusive rights granted by the government to inventors or their assignee to prevent them from commercial use of their invention. And when patent is granted for biological entities and for products derived from them, they are called biopatents. Primarily, USA, Japan, members of European Union are awarding the biopatents. बायोपिरेसी का मतलब होता है चोरी, मीन्स किसी के बायो रिसोर्सेज को यूज़ कर लेना जो कि अभी प्रॉपरली ऑथोराइज भी नहीं है और बिना पैसे दिए अगले बंदे को। मान लो मैंने कोई इन्वेंशन किया और मैंने उसको रखा हुआ, तो मैंने किसी को दिखाया ये देखो मेरा ये इन्वेंशन है, अगले बंदे ने उसको चोरी कर लिया, ना मुझे पैसे मिले ना मेरा काम भी चला गया, ठीक है। तो कुछ नेशंस जो होते हैं इसको रोकने के लिए कुछ लॉज डेवलप करते हैं, जैसे इंडिया में इंडिया पार्लियामेंट ने इंडियन पेटेंट बिल जो सेकेंड अमेंडमेंट में क्लियर किया था कि जो बायोपेरेसी के इश्यूज़ को दूर करता है। अब कंट्रोवर्सीज क्या हैं, क्या हुआ, बासमती राइस के साथ और टर्मरिक और नीम, तीन चीज़ों के साथ छेड़छाड़ हुई थी। जो राइस है इंडिया में कई सालों से है, एशिया की हिस्ट्री में है, इंडिया में ही 2 लाख वराइटीज़ ऑफ राइस आर देयर, ठीक है। बासमती which is a very nice kind of rice, it is unique for its aroma and flavor. It has 27 varieties cultivated in India. The American company Rice Tech, it won the patent on it in 1997, but originally it was of India. It just get patent. It biopraised, चोरी हुई. इंडियन फार्मर्स को तो पता ही नहीं था. They were unaware that what they are doing, why they are taking a all. Okay, so Indian basmati was crossed with semi-dwarf varieties and claimed as an invention. बिकॉज जो इंडिया की जो वैरायटीज थी वो थी सेमी ड्वार्फ वैरायटीज, उनके साथ उन्होंने क्रॉसिंग की किसी चीज़ की और अपना बना लिया, तो और पेटेंट उनका चला गया, तो इंडिया के बाहर से में नहीं आया. टर्मरिक और नीम भी ऐसे ही हुआ. अब यूज़ ऑफ टर्मरिक वाउंड हीलिंग में इंडिया में शुरू हुआ था, पर इसका पेटेंट किसके पास चला गया? मिसिसिपी के पास. मिसिसिपी मेडिकल सेंटर है 1995 में. एक और केस हुआ, ये जो फंगिसाइडल यूज़ है नीम ऑयल का, ये भी इंडिया में था, ये भी 1996 में चोरी हो गया था यूरोपियन पेटेंट ऑफिस की तरफ से. और अगर हमने इस पे ध्यान ना दिया तो हमारी रिच जो लिगेसी है, हम इतने अमीर हैं इंडिया में, सब कुछ चोर ले जाएंगे. सो वी नीड टू सेव, वी नीड टू एनकैश आवर रिच लेगेसी, वी नीड टू पे अटेंशन. So this was the whole chapter. Thank you very much. If you like the video, press the like button. Hit the bell channel.